next we have Dr. Arturo Casadevall. He is a physician and professor at Johns Hopkins. He's chair of the molecular microbiology and immunology department there. He is author of over 900 publications, member of many scientific societies, recipient of many honors, and he has a book out called What If the Fungi Won? And I am very curious to hear his thoughts on that matter. So I will turn it over to him.
Thank you. First, thank you for having me, and thank you to the previous speakers. I really enjoyed those presentations. I think the route to tonight is that I gave a talk. First, I wrote this book, then I've been getting invitations to talk about it, and I never actually put together a talk based on a book, but I went to the Advanced Physics Laboratory of Hopkins, and Daniel Berman was there, and he then contacted, I think, your prior president, and that's how I got invited. So that's the road here, and I'm going to share the screen, and hopefully, oops, what happened to my presentation again? All right, I'm gonna have to close it and open it again because this is inevitable.
No worries.
We had it working before.
Yep, I have faith.
Let me see if I can try again on share. Here it is. So, and let me project it. So what I'm a scientist, I'm used to giving scientific talks. So, but this one is kind of unusual in that what I'm doing is putting some highlights, some things in the book. So I just want to say before I start that that was an art that the Hopkins publisher found. But at first I didn't know what to make of it. But the more I look at it, the more I like it. Appears to be some human covered with some sort of fungus. So I thought I would talk about the world's largest organism, our closest relatives, a cold kingdom, fungal melanins, fungi and the origin of mammals, and fungi and global warming. So when I've been asked to talk to the media, this is one of the things they always want to talk about. This organism in Oregon forest that covers 3.5 square miles. It is estimated to weigh 35,000 tons and to be the oldest organism on Earth. And the reason that this is known to be so is because people have collected samples and essentially done DNA testing. And using DNA testing, you can see the diameter of this monster. You can also see it causes armillaria root disease. So some of the trees about it are sick. And people have asked me, are there bigger ones? And I think the answer is, I don't know. I don't know if anybody knows, because this kind of mapping requires a tremendous amount of sophistication and perseverance. So, and the other thing that people are very interested on is that our animals and fungi are each other's closest relatives. Now, this comes to a surprise to many, because many assume that fungi are closer to plants. But it turns out that when you do DNA analysis, we share about 50% of the DNA in homology. And this becomes very medically relevant, because if you try to make an antifungal drug, you want to target the fungus and not the host. But if the host and the fungus are so similar, that means that you have fewer targets. And that's part of the reason why in medicine we have only 3 major classes of antifungal drugs. Now, this is something that came to a surprise to me, and this is research from the pandemic. We had a thermal camera to to study melanins, and I'll tell you more a little bit about it in a couple of slides. So Radamés Cordero, who's in my laboratory, began to go for walks in the woods and took the infrared camera with him. And what he noticed is the fungi are quite cold. So that was known, in fact, having commented by people in the field, mycologists, that but what strikes me is look how blue they are. They are 4 to 5 degrees centigrade cooler than the environment. So we said, oh, that's really interesting. Does this extend to the microscopic fungi? So we went into the laboratory and got a bunch of fungal species and began looking at them. And to our surprise, the colonies are cooler than the agar around it. So this idea that the fungal world is colder than the environment applies to microscopic fungi. And here, what you can see on the right is going across a colony. And the interesting thing is, as you cross it, you can see that the edges of the colony get warmer, but then it drops rapidly. The colony is much cooler than the agar. This is even when you let these plates equilibrate. If you put them at 4 degrees, they still get colder than the cold room. So, and then we began to look at what was going on. And if you look carefully, you can see the water bubbles forming on top of the mushrooms and the colonies that form. And this is evaporative cooling. This is the same phenomenon that you have when you get out of the shower. The bathroom may be 100 degrees, but you walk out of the shower and you're still cold. What's going on there? Well, evaporation. So we began to have fun. And one of the funs to this is we made an air conditioning based on mushrooms. That's on the right here. And what you do is, we call it the myco-cooler. And I can tell you that it's the only patent application that I sent in and came back accepted without revision. I think the patent examiner looked at it and said, I've never seen anything like this before. But it was a relatively simple instrument in which you put the mushrooms inside a styrofoam box, and then you take a computer fan and you drive air through it, and it really cools. You can see look at the bottom. This is the red is how rapidly the temperature drops to the air. That's about 10 degrees centigrade. So that's quite a bit of cooling. And the idea is that as long as the mushrooms have water, they cool the air. And when they dry up, you can take them and you can cook them and you can eat them. We made the microcooler with stuff from the supermarket. I'm sure you can do better with some of those mushrooms that we saw with the first presentation. Now, this is something I learned that I'm fascinated about: fungal leather. Do you know that there are companies are using, you can grow the mycelia in the shapes of a hat, the shapes of a bag, and it's remarkable this thing can be done. I went to a meeting and I was so surprised at the creativity that can be used, and this will have a lot of advantages in contrast to plastics. It's biodegradable. You don't have to worry about it. And my hope is that, in fact, it takes off. There is a lot of efforts at using it for construction. That is, taking fungal mycelia and pressing it so that you can make it into walls. If you look into this in the internet, you will be surprised at the enormous amount of activity that is in this area. Now I'm going to talk to you about melanin. It's not melatonin, melanin. This is the pigment in your skin. This is what gives you color. And it comes from the Greek word melan, or black. It is found in all biological kingdoms. Trees make it, animals make it. Guess what? Most fungal species make it. In fact, even if you take a totally white mushroom and you open it up, you can find darker parts that are made of melanin. And it's associated with numerous functions. One of the fascinating things is that it is the only biological material I know of whether you can boil it in concentrated hydrochloric acid and recover it. In fact, that's how we make it in the lab. So the melanins are found everywhere. Here you see a banana at the bottom. The ink of a squid are melanin particles, and the shades of our skin is simply the number of melanin granules. And here's a fungus that I work with in the laboratory, Cryptococcus conformans. And if you give it L-Dopa, or you give it a substrate, the colonies become pitch black. The melanin is deposited on the outside, so it is in a position that can interact with the environment. And what I'm going to be telling you about is this melanin does a lot of things, but this is fungal armor. And to give you an example how we make it, if you take the yeast and you grow them in this material, like L-Dopa, they become black. And then you boil them in hydrochloric acid and you recover the ghosts. The DNA, the polysaccharides, everything is digested away, except these structures. And the way I like to think about this is looking at them with scanning electron microscopy. They are, in fact, composed of even smaller parts, which are melanin granules. And the way I think about it is that this is something between studded leather and chain mail with plates. It allows nutrients in, but it gives the fungus a tremendous amount of protection. You can, depending on the shape of the cell, you can recover melanins that follow the shape. You can see that in Aspergillus, they follow the shape of the spore. Everything is gone. This is just a hollow melanin shell. And in fact, we found an unusual shape, and we had it published in the New England Journal of Medicine as Images in Medicine, and I'm about to show it to you. It's a rubber duck. It looks just like a duck. But what you're really seeing is the original cell that is collapsed, and then the bud, it's itself budding. And that's just melanin. Everything else is gone. So what does melanin do? It protects against everything, from heat to enzymes, to your favorite oxidant, bleach, to heavy metals. And in medicine, it's a real problem because it protects against some of our most effective antifungal agents. But what I'm about to tell you is a story that of a very unusual properties of melanin. And it comes from, I'm very interested in a lot of things reading outside my field. And I read that we're going to go to the Chernobyl disaster. This is 1986. Chernobyl had a chemical explosion, not a nuclear explosion, but the chemical explosion, it led to a chain reaction, and the whole thing melted. And what the Russians did is they created a sarcophagus around it. This thing, by the way, as you heard from the first days of the war in Ukraine, still working. The Chernobyl reactor has 3 other units that are making electricity. But it looked like this structure was about to collapse. So the European Union was very worried that if this thing collapses, it will get all this dust that will go right into Europe. And they sent that robot in the middle. This robot went in, and so everything in there was black. And it wasn't because it was charred. It was coated with black fungi. The black fungi were growing on the reactor. They were growing everywhere. And this is a Russian report, and you can see some of the organisms that were recovered. There were Cladosporium species and a bunch of other things, all pitch black melanin. So the first thing we thought is, gee, maybe they're growing in there because the melanin is protecting them against radiation. So we studied the ability of fungal melanin. Sepia is the squid melanin to protect against radiation, and you get remarkable protection. You get, hold on a minute, see, my daughter is here. You get about 50% the protection with melanin that you get with lead. This is something that has led the NASA to be very interested on it because of the potential of using this in spacecraft, as I'm about to show you. So that was not the history, but here is something that could be very relevant to those who like dark mushrooms. We showed that just giving melanin in mushrooms can protect a mouse against lethal irradiation. So what you do is you take white mushrooms, black mushrooms, and you grind them up and you put them into a mouse. And then the mouse is lethally irradiated. Well, the ones who ate the black mushrooms survived. The ones who eat the white mushrooms plus synthetic melanin survive. And the ones who eat the white mushrooms die. What's going on here? Well, the melanin, the mushroom is carrying melanin. The melanin is sitting in the gut. The melanin is protecting the gut against radiation. So the animal basically does an oral transplant. And there's a lot of interest in this, obviously, because if there is a catastrophe, certainly we can get a significant amount of protection by consuming black mushrooms. And if you don't believe me, a group in India has pretty much shown exactly the same result that we were able to publish. So that was not the story. So even though melanin protects against radiation, the problem was that the fungi are very resistant to radiation. They don't even need the melanin. In fact, in the 1950s, the Atomic Energy Commission used to calibrate radiation fluxes by kill curves in Saccharomyces cerevisiae. So then the Russians published this other fascinating study. They show that the fungi like to grow towards a radiation source. Why wouldn't a fungus want to grow to a radiation source? Well, we began to think about it and spent a few years and showed that, in fact, the fungi can use radiation for food. Let me tell you a little bit about this. This was done in collaboration with Katerina Darachova. She is a nuclear chemist. And so we had the mushroom, the fungi, she had the chemistry and the radiation. And basically what happens is that the melanin allows the fungus to capture high energy radiation, that it can be converted into biologically useful energy. And you know, we were so excited about this, we sent it to the high-end journals, Nature and Science, and they reviewed it and they give us a lot more work. But in the end, the editors were queasy about accepting this. And after 2 years of trying, we finally got a final rejection in which we got 3 comments. The first one we really liked. It says this should be published because even if they're wrong, this is going to promote a lot of work. Reviewer number 2 said that we shouldn't publish it unless we can track an electron all the way down. And I remember saying, but you can't do that with photosynthesis. But reviewer number 3 really was the one who defeated us, because he or she wrote, I can find nothing wrong with the work. I simply don't believe it. So we were at the realm of religion. It was time to look for another journal. And the one thing I can tell you is that we published it in PLOS ONE that had in 2007 had just come out. And all the journals that rejected us had to carry this story in the news, but they didn't. So I don't know what you prefer. You prefer in the pages of the journal, but you can see the journals that rejected us. And one of the only criticism that really bothered me, my PhD is in physical chemistry, was that somebody said to me, Arturo, X-rays go through biological matter. What are you talking about? Gamma rays? Gamma rays are really, really energetic. How are they going to get through this stuff? What's going to stop them? This is a biological material. And we worked for about 5 years just trying to show that melanin could interact with gamma rays. And the proof came from Charles Turek at the Savannah River National Lab, in which he made a fungal melanin electrode. And he took the electrode, connected it to a voltmeter, and then put gamma rays through. And when he turned the current on, he got a current. So there was no way out. The fungal melanin was capturing gamma rays. And this work has now been confirmed by a group from the Naval Research Laboratory in DC. So it's out there, but I think for this group, when you see dark mushrooms, they're collecting energy and they are collecting it. And maybe the fungi have some capacity to make their own food. We don't think about the fungi, we think about them as degraders. We don't think of them as producers, but at least this work suggests that there may be a lot more to it. We also were intrigued, does the color of fungal colonies vary with latitude? And originally we thought that, near the equator, maybe the fungi near the equator will make more melanin because that will protect them against sunlight. It turned out to be the opposite. It was the fungi in the Arctic and the Antarctic that were pigmented. So pigmentation goes with latitude. And what was going on was, we in Cryptococcus neoformans, because you can feed it different compounds, they can make different color melanins. And then you can have a yellowish, a reddish type of melanin pigments. And then you can put in a light and you could see how quickly it warms. And the problem, and the reason for this was, the darker it is, the faster it warms. So the melanin was, in fact, we're collecting solar radiation. You don't want to melanize when you're in the equator because you're going to get too hot. But if you are near in the Arctic or the Antarctic, the pigment could give you the ability to harness heat. And we were able to show that, in fact, in a cold room, under the light of the cold room, the melanized fungi were able to grow faster because they were able to harvest the light that was falling on them, getting a few degrees warmer, and using that, both the warmth and maybe even the radiation for growth. And I leave you with two pictures. On the right are the Black Mountains of Antarctica, and the black comes from a lichen. And that's an alga and a fungus. The fungus is making melanin, and the mountain is totally covered with melanin. And I would argue that that melanin from the fungus is allowing harvesting of solar radiation. Now, the dark side of this, no pun intended, is that when glaciers begin to melt, the holes get filled of melanotic organisms. And then you get a vicious cycle by which the melanin warms up rapidly, and it leads to much more rapid melting of the glacier. This is something that NASA, NASA is very interested in the fungi. In fact, there's something called mycotexture. It's a program to think about how you can use fungi for space travel and to protect the spacecraft. Because think about it, if melanin protects against radiation, all you really need to do is bring a spore to the space station and grow it out there. And the idea would be that you can design spaceships in which the outer part of the ship, not maybe facing the space, you can grow melanotic fungi, and they will give a significant amount of protection. On the right there, you see a futuristic type of housing composed of mycelia that can repair itself for habitation in other planets and out in space. So the fungi may get us to other planets. NASA is very interested in this, and they have a significant research program that I would say that we are collaborating with, and we have some grants from them on this initiative. So melanin functions. In nature, melanin is important. It helps for virulence. It gives cell wall integrity. In the hospitals, antifungal resistance. In Chernobyl, it radio protection. And we think that it captures electromagnetic radiation. Industry and defense is very interested in this because equipment radiation shielding, personal radiation protection, bioremediation of contaminated sites. It turns out that melanin is better than activated charcoal. So in places that are contaminated with heavy metals and radiation, if you can grow some of the black fungi, they concentrate the pollutants. And then if you remove them, you're actually removing the heavy metals and solar energy capture. So I'm going to basically talk to you now about global warming and what's going to happen. So I think that most people will agree that global warming is a fact. Temperature is an intensive property. What that means is that once you raise by 1 degree, a cell, everything changes in the cell. Temperature affects all aspects of metabolism. And as the world gets warmer, all organisms, whether macroscopic or microscopic, must adapt or die. And global warming is going to have tremendous effects on the microbial world. I'm going to tell you about what hopefully is not ahead, but it's to give you a sense of the crisis that we are facing as humanity is facing. And that I want to take you back to 155 million years to the Permian. This is the greatest extinction event in the Earth history. 90 to 95% of all life on Earth ended. The trilobites, for example, had been doing fine for 200 million years. They ended. And the current understanding is that it occurred as over 80,000 years associated with climate change, and it was caused by increased CO2 and reduced oxygen. So how did that happen? There was certainly no industries back then polluting. Well, it turns out that there was a fire in what is now Siberia, in which a magma plume set a huge coal fire, a huge coal bed on fire. This burned for 80,000 years. It dumped all that coal into the atmosphere, and it basically changed the climate of the world, and it led to extinction. We are doing this in about 200 years. The Permian extinction took about 80,000 years for the coal thing to burn. So the other message is whenever the Earth does badly, the fungi do well. Look at this. This is this is geology separating the Permian from the world of the early Triassic. There is 1 meter of fossilized fungal spores in that mountain. So as the world of the Permian died, the fungi did well. They basically digested everything that was dying. And we can find evidence of that massive fungal proliferation in the geologic strata by 1 meter of fungal spores. I mean, you got to think about what could that have been like. All life that survived would have been exposed to massive amounts of fungal spores. And when you look at these fungal spores microscopically, they are very similar to plant pathogenic fungi of today. Now, you can't tell the species by looking at it, but it's been argued that fungi could have contributed to the crisis in the Permian by basically proliferating massively and putting whatever survived under tremendous degree of stress. So the effect of global warming, there are going to be 2 types of effects. Host-associated microbes, for example, the ones in your gut, we don't think are going to get affected that much. You maintain them at 37 degrees. But things out on the sidewalk, things out in the soil are going to have to adapt. And what's going to be the relevant stress is not the average temperature. It's going to be the number of really hot days. The number of really hot days, every day with the temperature goes above 100 degrees or so, the sidewalk is 140 degrees. And it's putting tremendous selection stress on all microbial life at the same time. So this is something that you all know, but the fungal kingdom is enormous. And it includes the major pathogens of plants, insects, invertebrates, and ectothermic vertebrates. Fungi are currently devastating entire ecosystems. The bats in North America, the white syndrome, we're looking at catastrophic amphibian declines from chytrid fungus, and another chytrid is killing the salamanders in Europe, and the snakes in North America are turning up dead from fungal disease. But here is what always, as a physician, what always interests me. Fungal diseases are very rare on mammals. People worry about bacteria, they worry about viruses, they worry about malaria. You don't hear them worrying about dying of a fungal disease. And to me, that was itself a very interesting question. Not only are they are rare, but relatively few fungal species are pathogenic for mammals, for humans. So here you have the host-associated one, they're modifieds, we all have them. Candida species, we have several, and pneumocystis species which can colonize individuals, especially children. And we get the environmental ones, things like histoplasmas, Pergillus, Cryptococcus, Coccidioides. But the point is, we're not talking about thousands of them. We're talking about a relatively few. In fact, if you know 10 fungal species, you know 95% of human fungal diseases. So not only are we very resistant, but we are susceptible to only a few. Here is the power of temperature. Daniel Smith is someone in my lab, and I'll tell you a little bit more. He's been collecting fungi in Baltimore. And when you take them, you plate them, they all grow very well at 30 degrees, but raise the temperature to 42 degrees, and only one grows. And that one happens to be Candida tropicalis, that is a Candida that is heat adapted, in fact, is a human pathogen. Before we had drugs, fungal diseases in the extremities used to be treated with heat. This is sporothrix shankai, and so an individual like this that would take the leg and basically put it on something hot corn presses, and just raising the temperature was enough to kill off the fungus and let the lesions heal. So let's talk about why mammals. When you drive around today, what do you see? Out in the countryside, you see mammals and birds. You don't see large reptiles. And you don't see large insects. Why mammals? So what bothers me about this is that the mammalian lifestyle is incredibly costly. I had 4 meals today. And I'm probably going to have, when I finish this, a few hundred more calories when I begin to have some wine afterwards. If I was a frog, frogs need about 1,000 less energy to get through the day. If we were reptilian, we only have to eat once a week. A crocodile needs to eat once a week. And yet mammals, how do we end up with mammals being the major animals that we see today, mammals and birds? Well, this dates back to the catastrophe that killed off the dinosaurs. And what I've been very interested in, how did this unfavorable lifestyle become dominant? And we now know, I just told you that invasive fungal diseases are extremely rare in mammals. Could there be a relationship between the fungi and us? And what I'm going to set you up is the theory that I've been arguing for 20 years, that we are here as a result of fungal selection at the end of the Cretaceous. So where's the origin of mammals? Mammals go back into the Permian, and they made it through that catastrophe. What are the advantages of you being warm? Well, you have a stable internal environment. Your heart doesn't have to adapt to outside temperatures. You are able to move and digest food during cold weather. You always see those pictures in Florida. Whenever they have a cold snap, the iguanas basically can't move and they fall out of trees. That's what happens when you can't regulate your temperature. I'm going to argue that it gives you tremendous protection against infectious disease and the fungi in particular. So what is the evidence that your high temperature protects against fungal disease? Well, when you look at the world today, what you see is that wherever the fungi are devastating ecosystems, they tend to be cold-blooded animals. Corals, they tend to be plants. In the turtles, Fusarium goes right through the turtle and kills the baby turtle. Frogs can be cured by putting them in frog saunas. This is a paper that was just published in Nature. There is a lot of interest on how to save the frogs. So one idea is you create a sauna and you put something warm. The frogs know instinctively that when they are sick to try to get warm, they go into these places, you raise their temperature, and they're able to get rid of the chytrid. Temperature is very powerful defense against the fungi. The human fungal diseases tend to happen in the cooler parts of the body. Nail fungus, athlete's foot. By the way, dandruff is a fungal disease. It's called Malasuisia species. and fungal diaper rash in babies in the much cooler parts of the skin. Experimentally, you can demonstrate this. This work done by John Perfect and others at Duke. You take a rabbit and you try to give it Cryptococcus neoformis, and you can't, even if you put it direct to the brain. But you can give it in the cooler parts of the bunny. You can put it in the skin, you can put it in the testes. But if you give steroids that are immunosuppressive, temperature is not enough, and you get a dead bunny. So this begins to suggest that our remarkable resistance to fungal diseases is the result of what we call adaptive immunity or advanced immunity and endothermy, higher temperature. So now the bats teach us an important lesson, and it goes totally consistent with it. So bats in North America were fine up until 2006. And then somebody went to a cave in New York, and they noticed the floor was covered with dead bats. And the bats had something growing in their nose, this white nose, and these are the hypha of this fungus. This fungus thought to have been introduced from Europe. And here is the interesting thing. The fungus grows well at low temperatures, but it cannot take high temperatures. So the bats are fine in the summer, but in the winter, they have to hibernate because there are no insects. As soon as they hibernate, their temperature drops to 10 to 12 degrees, and they become susceptible to this fungal disease. If you take the bats that are infected and you bring them into the laboratory and just feed them, just feeding them raises their temperature. They're able to clear the fungus. Now, this is a little bit more complicated, but I tell you what, it's actually simple. So I became very interested in this and talked to somebody in a collection and went to a collection and asked when you put a sample in a collection, what they often do is they check the temperature susceptibility. What is the maximal temperature that it grows to? What is the best temperature? And Vincent Robert basically is a bioinformatics guy, and he was able to get into the records and show that fungi do very well to 30 degrees, but above 30 degrees, most fungal species can't make it. So for every degree above 30 degrees, you can exclude 6% of fungal species. Your temperature is giving you tremendous protection. I told this to my friend Aviv Berkman, who's a mathematician, and he knew that there was a formula for calculating the cost of being hot. And what he did is he solved two equations simultaneously. He said, what will be the optimal temperature by which you get the most protection against fungi and you don't have to eat all the time? And it turns out that temperature is 37 degrees. Your temperature appears to be optimally selected to keep out the fungi without you eating all the time. You could be 41, 42 degrees. You will get a lot more protection against a lot of things, but you will probably have to eat 8 times a day. Bunnies have to eat all day long just to maintain that temperature. So let's see where we come from. I told you that our ancestors go back to the Permian, but things really take off 65 million years ago. 65 million years ago is the beginning of the age of the mammals. And what you can see in here is the Cretaceous, that is green, ends, and then you have the Paleogene, that's the end of the dinosaurs, and you have this enormous mammalian radiation that gives you everything from bats to humans to whales. The two groups of organisms who do well are the birds and the mammals. These are the major groups who did well after the calamity. And what I'm interested in is what could have selected for this energetically costly lifestyle. The way to think about it is you can see the dinosaurs end, the mammals take off, the birds take off, and somehow the crocodiles manage to make it. Some of the amphibians do. But just like at the end of the Permian, there was massive fungal proliferation at the end of the Cretaceous. So what killed the dinosaurs? Well, people used to say it was small mammals that ate the eggs. That makes absolutely no sense. Mammals were around, mammals were barely making it in the world of the dinosaurs. This one doesn't make any sense either, but I found it in the internet. No, they didn't die from smoking. They could have died in part from increased volcanism. There was a lot of volcanism 65 million years ago. and there is something known as the Deccan Traps. This is the magma plume that make the Hawaiian Islands. You look at them in a map, and they're in a line. And the reason they're in a line is because this plume opens up, makes an island, then you have plate tectonics, and the plates move, and then it opens up again and gives you another island. However, most people think that the big hit was an asteroid that hit what is today Yucatan. There are at least two other explanations out there. One of them has been published in a very prestigious journal, so I'm just going to share it with you, and you can think about whatever you want about it. Somebody calculated that they could have passed enough gas to cause global warming. I don't believe it, but look, it's out there, published in 2012. And the other explanation, I will not argue for, because as a scientist, I have no evidence for or against, and I certainly can't falsify it, but I'll share it with you in silence. So let's go back to what happened. It was 65 million years ago. The Earth was a warm, volcanic, forested planet. There were forests in what is now Antarctica. The Atlantic was tiny. The world was populated by the great reptilian megafauna. You have the Dinosaurs. You have all these enormous creatures. And then this rock, which is about 17 miles, hit what is now known as Yucatan. Next time you go to Cancun, you will see that there are people at the airport trying to take you to see the cenotes. The cenotes are these structures, which are some deep holes in the ground that connect to the Caribbean. And the Mayans used to do sacrifices there. I urge you to go, not because of the sacrifices, because they probably don't have anything to do with sacrifice, because that's the rim of the crater. The limestone melted, and when it cooled, created this fascinating geologic structures. So the planet got hit, and it was a calamity. In a matter of hours, it was devastation. Mile-long tsunamis raced through North America. And this warm, forested planet, the forest came down, and it cooled about 12 degrees in a matter of a day. 12 degrees centigrade. That's a tremendous cooling. And there is evidence for massive fungal proliferation, because you now had no sun, because the smoke and the dust had been kicked off. The temperatures dropped off. It was, perfect for fungi. Dead vegetation, cool, moist, and no sunlight. And the argument is that whatever survives the calamity would have been exposed to massive fungal inocula. Amounts of spores would have been unbelievable. And about 20 years ago, I proposed that there had to be a connection between this. I remember I wrote this like kind of tongue-in-cheek, and over the years continued to work on it. So it goes like this. The reptilian megafauna is killed by the impact blast, the tsunamis, everything. Maybe fungal diseases contributed, but you don't need to. But reptiles, we know we have reptiles today. Some reptiles made it. So reptiles fight off fungal infections with induced fevers, but there was no sun. Reptilian lay eggs, but these were susceptible also to fungal diseases. And in fact, today, you can still see dinosaur eggs that have been invaded by fungi that will kill the baby dinosaurs. So the question to me was, clearly reptiles survive, clearly mammals survive. Why didn't we have a second reptilian age? How do we end up with mammals and birds? And this is an idea that a fungal filter at the end of the Cretaceous selected for endothermy. Endothermy is a fancy way of saying warm-blooded. Birds, mammals. And the idea is very simple. You had survivors, or you had both reptiles and small mammals, and then you had this fungal bloom, you had this devastated planet, but the mammals with their internal combustion engines were able to move, were able to forage, they were able to eat mushrooms, and their populations just became much larger. So when the skies cleared and the planet recovered, you just had a lot more mammals than you had reptiles. And this became the founder populations. And by sheer numbers alone, they gave us the great mammalian radiation, of which we are one of the species that came out of that. I continue to write about this and update it. I like to read the literature and the geologic literature, and these things need to be updated. I will tell you that somebody told me I had to give it a name because everything in science is a name. So they came out fungal infection mammalian selection of FIMS. I haven't been challenged yet. There are two possibilities. One is nobody cares. So no challenge. The other possibility is, nobody's figured out how to push against it. I mean, I've been publishing these ideas in peer-reviewed journals, and I'm surprised that I haven't been challenged yet. It is internally consistent. It provides a selection mechanism for an energetically costly lifestyle. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease. It argues that your temperature is a major defense mechanism. And it also explains why today, when you get a fever, the first thing you do is you take Tylenol. Because today, when we get diseases, they tend to be primarily organisms that are already temperature adapted. But you go out there mushroom hunting, and you fall in the forest, and you slash your leg. The organisms that are coming in are not temperature adapted. And for those organisms, a couple of degrees of fever could give you tremendous protection. So what's going to happen in climate change? Well, today, most pathogenic fungi are near the equator. But we know that fungi can be very rapidly evolve in the laboratory for higher temperature. What you see on the right side there is a report from a company who had a fungus to kill insect pests. But the insects learned to sun themselves and kill the fungus. So what they did is they were able to by just basically taking the organism and raising the temperature every couple of days, in a matter of several weeks, they were able to train it to grow at 36 degrees. We already seen that the fungi are adapting. When you compare species from cities and from the rural areas, the species from cities have greater thermal tolerance. They grow better at higher temperatures. Cities are heat islands, and they are often as much as 5 degrees centigrade warmer than the rural areas around it. In fact, we have documented this in Baltimore. We went to cold neighborhoods, cooler neighborhoods, and warmer neighborhoods. And the remarkable thing is that within one city, the fungal species from the cooler neighborhoods were less thermally tolerant than the ones from the hotter neighborhoods. So the fungi are adapting to temperature every summer. And we're continuing to work on this, and we think that this is tremendously concerning. This is some work that we did a while ago. It's messy because we went to the collection and we asked a simple question. When things are deposited per year, what is the maximal temperature? The bottom lines are the ascomyces, the top line, no, I'm sorry, the basidiomyces that tend to be more susceptible, less temperature resistant. The top one is the ascomyces. You can see the ascomyces lines are flat. The basidiomyces are increasing. That suggests that the fungi have already been adapting for several decades. So with that, in 2010, we wrote this paper warning people that global warming will bring new fungal diseases for mammals that are unknown. And the idea is simple. You are protected by your temperature. That's keeping out most fungi. But what happens if the fungi adapt to higher temperatures? There are a lot of stuff out there that can cause disease. And then you can see in the New York Times, a mysterious infection spanning the globe in a climate of secrecy, the rise of Candida auris. Candida auris is a fascinating situation. It was unknown to medicine till 2007. Physicians had never seen it before. They recovered it from the ear of a patient, and they call it auris, Latin for ear. And then people say, well, maybe we missed it. So they went into all these collections, and they found an occasional isolate in South Korea, Japan, and in Pakistan. But here is what's remarkable. Then, within a period of 4 years, it appears simultaneously in 3 continents and begins killing patients, in primarily immunocompromised patients. What's going on here? South Africa, Venezuela, and India are very, very different climates, very different cultures, very different agricultures, and yet these organisms have emerged. And they are different. It's not like somebody took a plane and spread this infection. These are very, very different clades. So clearly we have a new pathogenic fungi. How did it emerge independently in 3 continents? So the 3 theories of its emergence. It was there all the time, but people missed it. Problem is, people have gone and done a retrospective analysis, and it was not there. It was a consequence of the overuse of azoles. These are fungicides in medicine and agriculture. Again, it's not clear how antibiotic selection will give you virulence. Antimicrobial resistance and virulence are different. And then we have argued that this is the first example of a fungus breaking through or thermal defenses. The problem is conjecture and circumstantial evidence. So what is the evidence? Well, the best evidence to me is that it emerged simultaneously at the same time, and you can't find a common denominator in these areas except that they all got warmer. Then we'll compare them to the closest relatives. The closest relatives are not temperature adapted, though the other Candida relatives. So Anuradha Chaudhary in India was able to find it in the environment. He was able to find it in the Andaman Islands. He found it in 2 places, a marsh that is remote and nobody goes to, and a beach that is frequented by people and where they may have brought it in. The isolates from the beach are temperature adapted. The isolates from the marsh are almost there, but they struggle to grow at 37 degrees, almost telling you that these things are adapting and probably are adapting very rapidly. And here is the next problem. We are getting colder. Yes, humans are getting colder. How can that be? Well, this study went and looked at temperatures over a century. And temperature is something we can measure. We can't say, well, we didn't know how to measure temperature in the year 1900. We have been able to measure temperatures at the beginning of the scientific revolution. But what is happening is that we're about 1 degree colder now on average than we were, or our great-grandparents were. And the thought here is that they lived in a dirtier world where they had more infections. And infection produces inflammation, and inflammation produces heat. So in our cleaner world, we may be getting colder. Now, humans are not all 37. Some of them are 35, some are 36, some are 38. Some of them run hot, some of them run cold. So you can imagine what I'm concerned about. The fungi are adapting to higher temperature, and some humans are getting colder. And I think that it's a collision to come. Now, to add more concern is that temperature itself tends to cause mutagenesis in the fungal world. And this is now being documented. This is the work of Asiya Gusa at Duke. And what she showed is that when you increase the temperature, you mobilize all these genetic elements that basically cause all kinds of heterogeneity. And they in some cases, they enhance virulence. In some cases, they lower antifungal susceptibility. So temperature is not only adaptation to being warmer, but it could be giving us creatures that are, in fact, different than we know them as a result of increased mutagenesis. I'm almost at the end, but I can't resist talking about this, and everyone here probably has heard about it. I had no clue till I began to get emails about this because people said, did you advise them? So as you know, the story is that some cordyceps basically adapts to temperature and is able to cause disease in people, turn them into zombies, and then you had this zombie apocalypse. And all the reporters and all this call me, and always the question is the same: could this really happen? And my answer is always the same: it's improbable, but not impossible. It's improbable because we certainly don't know of any cordyceps or fungus that can turn a mammal into a zombie, but not impossible because guess what? Some fungi make LSD, and some fungi make hallucinogens. So how do you know that there isn't some fungus out there that can do something to your brain? Thank you very much. I hope you enjoyed the tour.
Thank you so much. One of the questions that occurs to me, you were using the example of Candida aura in places like India, where presumably the ambient temperature is above human body temperature sometimes. So, like, how does it survive in the environment? Do we have a sense of that?
Well, it's already adapted. So when you recover them, you know, a lot of them, the soils get cool pretty quickly. So if you go down a couple of inches, or if you put it in shaded areas, they basically just have to often make it to the evening. But I think the message is that this kingdom is very adaptable. And even though a lot of the mushrooms that people in the audience love are going to get into trouble with temperature, the microscopic ones may do well. And they're the ones that cause disease in us and in plants. And I think this is a lot of trouble ahead.
Mm-hmm. Do other folks have questions? You want to put them in the chat? Let's see. April's asking. Oh, now my cursor not working. I'm curious how they date. Oh, this goes back to the humongous fungus at the very beginning of your presentation. You talked about how old it is. Do you know how they know how?
I think the estimate is from a growth. Fungi don't age, by the way. The idea is by how much is spreading as a function of time. So you look at it, what is the spread, and then you trace it back to a center, and you say how many years would it have taken to get here.
Mm-hmm. That makes sense. Okay, so Eric's asking a question along the lines of what I was thinking. We have the impression that fungal infections are relatively greater near the equator, but there's heat, but maybe that goes with it, that the fungi near the equator are adapted to the heat.
Exactly. Statistically, you just have more of them that are adapted, and that's why we get more of them. That's why we have more presumably fungal diseases near the equator.
That's so interesting. Yeah. Anyone else have questions? I know we're coming right up on 9. So we've made a recording of this. We'll put it up on YouTube for folks if you want to go back and watch it again. Oh, here's one. Agnes, who I know works at the NIH, is asking, are there any new fungal therapeutics in the pipeline?
So the answer is, Agnes, yes. There are some that are coming through. There are only 3 major classes or best antifungals. Drug was discovered in 1957, the year I was born, and that's still the workhorse. But there are some that are in the pipeline. The big problem in treating fungal diseases is that whereas if you have a bacterial disease, you can treat it in a couple of days, maybe a couple of weeks. Fungal diseases often take months to years. People have to be treated a long time, and we don't really understand why that is. But it just happens to have to be very resilient organisms. Once they get into tissues, getting them out of there is really, really hard. So hopefully there will be more antifungal agents.
You mentioned some of the field work that folks in your lab are doing around Baltimore. If you ever want help down from the DC area or want
Actually, I was thinking the same thing, because clearly you guys are seeing a lot of stuff on it, but at least maybe this talk will give you some awareness or something to keep in mind. You know, I noticed the first speaker, the mapping, to begin to think about areas. By the way, you can get thermal imaging of Washington and the area, as to whether some of these changes that you're seeing in the prevalence of the fungus reflect changes in temperature.
Yeah, yeah.
And that could be a very important way in which you guys could do citizen science that nobody's doing. And it could provide some very interesting insights that the community could contribute to.
That's a really interesting idea. Chris Wozniak's asking, is there any increase in fungi which infect both plants and animals?
Yes. So it turns out the fungi are trans-kingdom pathogens. So Fusarium, for example, Fusarium species will kill plants, kill people, kill amoeba. they basically are remarkable in that they are non-specific. You know, HIV, you can only get HIV humans. You can't give HIV to your dog. But these things are remarkable. And once they get into a host, they simply don't care. They, their ability to cause disease, to take down a plant, to take down a mammal is just remarkable.
It's a little scary. Yeah. All right. Not
Well,
seeing.
if anybody's interested,
Yeah.
and we can maybe talk more about it, about doing some citizen science on worms and things like that. I think that we're about to go through another really hot summer. In fact, we're going to get really hot in a couple of days again. Think about what that's doing to the natural environment. And, you guys have been collecting data over many ways, over many years, and try to correlate that with some of the climate changes that we're seeing. Could give us some hint as where things are going.
Yeah. Yeah. There was a member of our club who kept little notebooks for years of the mushrooms that we were seeing on forays, and I don't think they've ever gotten, digitized in any way that's usable. We'll have to see if we can track some of that stuff down. Because we've been using iNaturalist in more recent years for people to record their observations, and that's becoming a really rich resource. But it doesn't go back as
but
far.
it'd be nice to correlate that, right, with, because the temperature of the area is very well known. I mean, it's recorded every day over it. If those databases could be kind of linked, there may be some very interesting. The mushrooms, the fungal world is hypothermic. It likes lower temperatures. It's colder than the environment. So it's going to be very, very susceptible to whatever is going on with climate.
Yeah. Yep. Oh, and Agnes is saying, so we have this one annual retreat in over Labor Day, although it's moved dates over the years, but Agnes is saying we could get historic temps for that spot where we've been collecting, in exactly the same spot.
Same place, yeah, and see
I
if
think
there's
we've
a
got
correlation.
list for each year, yeah.
You could all have a communal paper.
Right. $900.
Whoever writes it goes first, and everybody goes alphabetically.
That's great. Well, thank you again so much. And if you have other ideas of little citizen science projects that would be fun for us to take on, feel free to shoot us an email.
Well, I will, now that I'm kind of connected, and similar, the same thing, you know how to reach me.
Yeah.
It's a pleasure to meet you, and I had a really, really enjoyable night listening to the prior presentations. Thank you for having me.
Yeah, thanks for coming. All right, take care,
Bye-bye.
everybody. We'll see you in August. Good night.